Good evening. I'll present my screen. Then we can start now. Can someone kindly unmute and let me know if my screen is visible and if I'm audible? Yes. Am I audible? Hello. Are you saying something I'm not able to hear? Can you guys see my screen? Hello. Can you guys see my screen? Wait, I cannot hear anybody. Don't know why. Let me check if my screen is visible. Sorry, I don't know why I'm not able to hear you guys. Yes, okay, great. I'm audible. Just check if I'm visible also. I'll just check this. Okay, so my screen is visible. Perfect. Yeah. So I'm guessing this. So my screen is visible. So let's start with today's session. Okay. So good evening, everyone. My name is Aishi, and I'm a second year PhD student from the Department of Chemistry, IIT Bombay, and I'm your PMRF. I'm a PMRF research scholar and a NPTEL TA for this course. For this course, Applied Environmental Microbiology. So, as you know, that every week we have a live session where we discuss some of the sample problems from either this year's assignments or past year assignments, and we discuss it to the depth so that it will help us solve the current year's assignments in the future. So we'll start today's assignment discussion. Today's sample problems discussion. So let's start the question that we have for today. So the first question that we have is, which of the following processes involves the removal of suspended solids from water? Disinfection, sedimentation, filtration, chlorination, coagulation and flocculation. What do you think is the correct answer? Anybody? Okay, just unmute and let me know, because I won't be able to go back to the Google Meet screen every time and see the messages, the chats. So if someone can unmute and let me know. Which of the following processes involves the removal of suspended solids from water? Anybody? Okay, somebody has written something. I'll have to go back and check. Yeah, correct, sedimentation. So if you can unmute yourself, if there's no issue with your mic or something, maybe you can unmute and let me know. So yes, sedimentation. So the first thing that clicks in my mind when I see suspended solids, the first thing that will click in my mind will be gravity, and from that I can easily go towards sedimentation. So we'll learn what exactly is sedimentation. It's nothing but a physical water treatment process that involves the settling of suspended solids in water. So when water containing suspended solids enters the sedimentation tank, the velocity of the water flow is slowed down, and this reduction in the velocity allows gravity to take over, and the heavier suspended solids settle to the bottom of the tank. The sedimentation tank is designed to provide a large surface area for the settling of suspended solids, and the size and shape of the tank as well as the flow rate of the water are very carefully controlled to ensure optimal sedimentation. So as the suspended solids settle, they form a layer of sediment which is periodically removed from the bottom of the tank. So everything boils down to gravity, as I said. So as the suspended solids will start settling due to them being heavy, they'll form a layer of sediment which is periodically removed from the bottom of the tank, that is cleaned. And the settled solids or sludges are typically removed from the sedimentation tank using a scraper mechanism. So the scraper moves slowly along the bottom of the tank, pushing the sludge towards the central collection point where it gets removed from the tank. And the clarified water, as you can see over here, the clear clarified water, which is now free of any kind of suspended solids, uh, this clarified water is then discharged from the tank for further treatment. So I think this is one of the preliminary steps of treating household or treating any kind of water, because this is very simple. You just let it settle for a time being, and then you can scrape out the dirt from the bottom. Okay, and the overall sedimentation is an effective and very widely used method for the removal of suspended solids from water, and it is particularly useful for treating large volumes of water, such as in municipal water treatment plants or industrial processes. And the main purpose of sedimentation is to enhance the filtration process by removing the particulates. So sedimentation is a process by which the suspended particles are removed from the water by means of gravity separation. So in this process, the water will pass through a relatively very quiet and still basin, and in these conditions the flock particles tend to settle to the bottom of the basin, while clear water passes out of the basin over an effluent baffle or weir. And this schematic basically illustrates a typical rectangular sedimentation basin, and the solids collect on the basin bottom and are removed by a mechanical sludge collection device or a scraper, and the sludge collection device then will scrape the solids to a collection point within the basin, from which it is pumped to disposal or to a sludge treatment process. And this process of sedimentation involves one or more basins called clarifiers. So clarifiers are relatively large open tanks that are either circular or rectangular, like this one in shape, and in properly designed clarifiers, the velocity of the water is reduced so that gravity is the predominant force acting on the water solid suspension. So when velocity is reduced, gravity will be the most the gravity towards the bottom will be the most enhancing prominent force acting on the water solid suspension. And the key factor in this process is speed. So the rate at which a flock particle drops out of the water has to be faster than the rate at which the water flows from the tank's inlet or slow mix to the outlet or filtration end. And the difference in specific gravity between the water and the particles causes the particles to settle to the bottom of the basin. Now some plants have added baffles or weirs in their sedimentation basins to limit short circuiting through the basins, promoting better settling. And other forms of sedimentation used in the water industry can also be named, such as tube and plate settlers, solids contact clarifiers, sludge blanket clarifiers and contact clarifiers, dissolved air flotation, and all this. So as you can see, the sedimentation process will be very effective in removing suspended solids in industrial wastewater, and clarifiers, which will be rectangular or circular, will be most commonly used in the application of sedimentation in wastewater treatment facilities. And although this process sedimentation is a method of treatment used in almost all domestic sewage, like almost in all domestic sewage treatment plants, it should be considered for industrial waste treatment only when the industrial waste is combined with domestic sewage or contains a high percentage of settleable suspended solids. Now, the efficiency of the sedimentation tanks depends on the following factors such as retention period, wastewater characteristics, tank depth, flow surface area, overflow rate, operation temperature, particle size, inlet and outlet design, etc. And another consideration in the design of sedimentation process is the characteristics of the suspended solids. So small and very difficult to settle particles in suspension can be flocculated and buoyed to the liquid surface by the lifting power of many minute air bubbles that attach themselves to the suspended particles. And floated agglomerated sludge can be readily and continuously removed from the surface of the liquid by skewing. And screening of industrial waste is generally practiced on waste containing larger, suspended solids of variable sizes, such as from canneries, pulps, and paper mills or poultry processing plants. And it is very economical and an economical and effective means of rapid separation of these larger suspended solids from the beginning, from the remaining waste material. So this is the entire process of separating suspended solids from water by allowing them to settle under the influence of gravity, and this is extensively used to remove impurities from water before it is discharged into lakes or oceans. And as I already mentioned, the basic principle is that the velocity of the water flow is reduced to allow the heavier particles to settle to the bottom of the tank or basin, and the suspended particles can include organic matter, clay, sand, silt, and other substances that are present in the water. And these particles can cause cloudiness, taste, and odor problems if the water is left untreated. And these are carried out in large tanks or basins specifically designed for this purpose, and the tanks are rectangular or circular in shape and are typically made of concrete or steel. And the water flows into the tank at one end and flows out at the other end. So as the water enters the tank, it slows down, allows the heavier particles to settle to the bottom of the tank. The settling process is aided by the use of chemicals called coagulants and flocculants, which cause the suspended particles to clump together into larger particles that are easier to settle. And then the settled particles form a layer of sediment on the bottom of the tank. It is periodically removed using a scraper mechanism. Like what happens is that the scraper slowly moves along the bottom of the tank and pushing the sediment towards the central collection point where it is removed from the tank. And the clarified water, which is now free of any kind of suspended solids, is discharged from the tank and sent for further treatment. See, and the sediment that is removed from the tank is typically sent for disposal or any kind of further treatment depending on the nature of the particles and the local regulations. So that is why this is a very widely used method for the removal of suspended solids from water, particularly used for treating large volumes of water like municipal water. And it is relatively simple and inexpensive, and it can be used in conjunction with other treatment processes such as filtration, disinfection, to provide high quality drinking water. So that is what sedimentation is all about. Moving on to the next question. Which of the following statements about antibiotic resistance is true? Anybody? Anybody? Is anybody written anything out there? I can see. Option 3. Okay. Let's see what is option 3. Yeah, right. Overuse and misuse of antibiotics contribute to the development of antibiotic resistance. So that is the correct answer. So let's discuss a little bit about antimicrobial resistance. Sorry, we've already talked a little bit about this topic in the last few classes. So antimicrobial resistance is basically a huge challenge because it poses a significant threat to human and animal health. And antimicrobial resistance occurs when microorganisms such as bacteria, viruses, fungi, parasites develop resistance to the drugs that were previously used to treat them. The drugs they were previously susceptible to, like vulnerable to, the drugs that could kill them previously. And this makes it harder to control these infections. And in severe cases, it can even result in prolonged illnesses, increased morbidity and mortality rates, and increased healthcare costs. So, antimicrobial resistance can also lead to the spread of infectious diseases, as infected individuals are less likely to be cured or effectively treated, and therefore more likely to transmit the disease to others. And it can also result in the emergence of new infections that are difficult or very impossible to treat, leading to public health emergencies. And furthermore, antimicrobial resistance is a global challenge that requires a coordinated response from all the major healthcare professionals, researchers, policymakers, and the public. And without such a response, the problem is likely to worsen, and more people will be at risk of becoming infected with the resistant strains of microorganisms. And finally, antimicrobial resistance can affect many aspects of society, including food, security, animal health, and the environment. It's a complex problem that requires a multifaceted approach to address effectively. Okay. So, as I said, it's a natural process that occurs when bacteria evolve to become resistant to the antibiotics used to treat them. However, overuse and misuse of antibiotics accelerate this process, making it more difficult to treat the bacterial infections. So, when antibiotics are used excessively, unnecessarily, or inappropriately, they can kill some bacteria while allowing other bacteria to survive and reproduce. And these surviving bacteria, which can then be passed on to the other bacteria through horizontal gene transfer, as a result, the number of antibiotic resistant bacteria increases, making it more difficult to treat these infections. So what happens is that at some point of time, even if you're keeping on giving the drug, at some point of time, the bacteria will figure out a mechanism to inhibit the drug or stop the action of the drug. And then once it gets resistant to the drug, it can then actually transfer this quality to other bacteria in the culture using horizontal gene transfer. So that is why more and more bacteria with this multi-drug resistant property is coming up into existence. And additionally, when antibiotics are used to treat viral infections such as the common cold or flu, they are very ineffective, as antibiotics only target bacteria to antibiotic resistance, and by basically it only targets bacteria, and using antibiotics for viral infections can contribute to antibiotic resistance as well by promoting the growth of resistant bacteria in the body. So if you have a viral problem and you go and get some antibiotics which kills bacteria, and you take the dose without consulting any doctor, you are bound to give rise to some kind of microorganisms that will develop a very prominent prominently it will become susceptible to any kind of bacterial infections. So, and moreover, when antibiotics are prescribed at subtherapeutic doses for too short a duration, or patients do not complete the entire prescribed course of antibiotics, and it can lead to the survival of bacteria with mutations that make them resistant to the antibiotic. And the overuse and misuse of antibiotics in agriculture and food products can also contribute to the development of antibiotic resistance. So, antibiotics may be used in livestock to promote growth or prevent infections, leading to the development of antibiotic resistant bacteria in animals. And these resistant bacteria can then be transmitted to humans through direct contact with animals, or during the consumption of contaminated meat. And overall, users and misuse of antibiotics select for resistant bacteria and provide a selective, contributing selective basically pressure for the emergence of resistance, thereby contributing to the development of antibiotic resistance. So that is what generally happens in this case. And antibiotics kill help our bodies because they are killing all types of bacteria. Some of the bacteria that make us sick get better at defending the antibiotic. The complete dose is not fulfilled. Some bacteria will have, they'll develop this property to defend drugs, and then that will give rise to the resistant bacteria that to multiply, making our generations less and less, effective. So this is how is an example of how antibiotic resistance spreads. So animals get antibiotics and develop resistant bacteria in the gut. George gets antibiotics and develops resistant bacteria in the gut. So it's pretty much the same. Even if you supply antibiotic, they'll get resistant bacteria in the slave, in his gut. And then drug resistant bacteria can remain on the meat from animals, or when not handled or cooked properly, the bacteria can easily spread to humans. And then you can see in the human excerpt, George stays at the home and the general community and spreads resistant bacteria. And George gets nurse care at a hospital, nursing home, or inpatient care facility. And in case of bacteria, fertilizer or water containing animal feces and drug-resistant bacteria is used on food crops. And drug-resistant bacteria in the animal feces can remain on crops and be eaten. So these bacteria can remain in the human gut. So these are all the techniques of antibiotic resistance. So you might have wondered, how does this antibiotic resistance occur? So there are many processes or techniques that can help in the development of antibiotic resistance. That is the ability of the bacteria to survive and multiply even in the presence of antibiotics, and that would normally kill them. And it can spread through a variety of mechanisms, including mutation. So bacteria can develop resistance to antibiotics through random mutations in their DNA and allow them to basically evade the effects of the drug. As you can see from here, this mutation has led to a modified drug target. So now the drug, even if it comes from outside, it cannot bind to the target site because the bacteria has changed the conformation of the drug target basically. Bacteria can also develop resistance to antibiotics through random mutations in their DNA that allow them to evade the effects. I've already talked about this. Horizontal gene transfer can be also a reason. Bacteria can actually acquire resistance genes from other bacteria through mechanisms such as conjugation, transduction, and transformation. And this means that the bacteria can share genes that code for antibiotic resistance, allowing them to quickly spread resistance to other bacteria. Then there comes overuse and misuse of antibiotics. So when antibiotics are used too frequently or inappropriately, it creates an environment that favors the growth of resistant bacteria. And this can occur when antibiotics are used to treat viral infections, which are not affected by antibiotics at all. So don't have this information. So this can occur when antibiotics are used to treat viral infections, or when antibiotics are prescribed unnecessarily for conditions that could be traveled without them. Also, travel and trade. This is also very important. The spread of antibiotic resistance. Bacteria can be facilitated by international travel and trade, and resistant bacteria can be carried in the gut flora of people or animals and can be transmitted to other places or animals in a new location. Also, agricultural use of antibiotics. Antibiotics are often used in agriculture to promote growth and prevent disease in livestock, and this can actually lead to the development of antibiotic resistant bacteria in animals, which can then spread to humans through consumption of contact with the animal feces. So overall, the spread of antibiotic resistance is a complex and multifactorial process that involves a combination of genetic, environmental, and behavioral factors. It is a growing concern in public health as antibiotic resistant infections are becoming more and more difficult and more costly to treat. So we'll move ahead to some of the different mechanisms that might work together to confer resistance to a particular antibiotic. Till then, does anybody have any question? We'll move on to the procedures for antibiotic resistance. But any questions, anybody? So we'll move to the questions. The next part. Yeah, so we'll move to the next part. Yeah. So these are some of the very prominent mechanisms that have come into light that may work together in combination, or maybe they are working together separately, or working separately to confer resistance to a single antimicrobial agent. So the first strategy is preventing access. So antimicrobial compounds almost always require access into the bacterial cell to reach the target site where they can interfere. That's the cursor. Yeah, and they can interfere with the normal function of the bacterial organism. Okay, organism. So pore channels are the passageways by which these antibiotics would normally, come inside, cross the bacterial outer membrane of gram-negative bacteria. Now, some bacteria protect themselves by prohibiting these antimicrobial compounds from entering past their cell walls. For example, one variety of gram-negative bacteria reduces the uptake of certain antibiotics such as aminoglycosides and beta-lactams by modifying the cell membrane protein channel frequency, size, and selectivity. And prohibiting entry in this manner will prevent these antimicrobials from reaching their intended targets, and that for aminoglycosides and beta-lactams are the ribosomes and the penicillin binding proteins respectively. So when the drug is not allowed to enter inside the cell only, it will prevent the antimicrobials from reaching the intended targets. That for aminoglycosides and beta-lactams are the ribosomes. So these two kind of family of antibiotics target the ribosome of the bacteria, while the penicillin binding proteins are taken care of by the beta-lactams. So when you are not letting them enter the cell only, then there's no point, because resistance can be very easily maneuvered. And this mechanism can be observed in many species such as the Pseudomonas aeruginosa. It has resistance against the carbapenems, which are beta-lactam antibiotics. Then there is Enterobacter aerogenes and Klebsiella species against carbapenems. Then there is vancomycin intermediate resistant S. Staphylococcus aureus, or VISI strains that with thickened cell wall trapping vancomycin. So many gram-negative bacteria against aminoglycosides, and many gram-positive, gram-negative bacteria against quinolones. So this strategy is basically used by these types of bacteria. We'll move on to the next strategy, which is eliminating antimicrobial agents from the cell by expulsion using efflux pumps. So to be effective, the antimicrobial agents must also be present at sufficiently higher concentration within the bacterial cell. So some bacteria may possess membrane proteins that act as an export of or efflux pump for certain antimicrobials, extruding the antibiotic out of the cell as fast as it can. So this results in low intracellular concentration and that are insufficient to elicit an effect. And some efflux pumps selectively extrude selective specific antibiotics such as macrolides, glycosamides, streptograms, and tetracyclines, whereas others referred to as a multiple drug resistance pump, a variety of structural diverse anti-effectives with different modes of action. And this particular strain, where this particular the particular bacteria that shows the example of intake of outside materials by the process of using the help of influx pump is something that E. coli and Enterobacteriaceae against tetraclines also use. So interbacteriaceae against chloroform is also possible. So the Staphylococci against macrolides and streptogams, and the Staphylococcus aureus and Streptococcus pneumonia against fluorocones. So these are the two basic main strategies behind antimicrobial resistance. Also, did you know that the influx pumps are variants of membrane pumps possessed by all bacteria? So both pathogenic and non-pathogenic to move lipophilic or amphiphilic molecules in and out of their cells. And some influx pumps are used by antibiotic producing bacteria to pump antibiotics out of their cells as fast as the antibiotic is made, and this constitutes an immunity response mechanism for the bacteria to prevent being killed by its own chemical weapon. So that is what happens in case of efflux pump. Then we'll move on to the third strategy, which is inactivation of antimicrobial agents and modification via degradation. So what happens in this case is that another means by which the bacteria can actually preserve themselves by destroying the active component of the antimicrobial agent. So a classic example would be the hydrolytic deactivation of the beta-lactam ring in penicillins and cephalosporins by the bacterial enzyme called beta-lactamases, and the process inactivates penicilloic acid, causing it to be ineffective in binding to PBPs, thereby protecting the process of cell wall synthesis. So that is what is the third mechanism that they follow, and this strategy has been found in Enterobacteriaceae against chloramphenicol, gram-negative and gram-positive bacteria against aminoglycosides, and all this. And did you know that the first antibiotic resistant mechanism described was penicillinase? It was first reported by Abraham and Chain in the article, "An enzyme from bacteria also destroys penicillin." So this is the third strategy that we have that people generally use. And less than 10 years of the clinical production of penicillin resistant Staphylococcus aureus was observed in a majority of gram-positive infections in people. And the initial response by the pharmaceutical industry was to develop beta-lactam antibiotics that were unaffected by the specific beta-lactamases secreted by the Staphylococcus aureus. However, as a result, bacterial strains producing beta-lactamases with different properties began to emerge, as well as those with other resistant mechanisms. And this cycle of resistance, counteracting resistance, continues even today. And beta-lactamase inhibitors from laboratory to clinic. So that is how this entire process works out. This is the third strategy, which is the inactivation of antimicrobial agents via modification or degradation. There is another strategy, which is the modification of the antimicrobial target. So some resistant bacteria evade antimicrobials by reprogramming or camouflaging critical target sites to avoid recognition, and therefore, despite the presence of an intact and active antimicrobial compound, no subsequent binding or inhibition will take place. And this strategy has been seen in Staphylococcus against methicillin and other beta-lactams. Then Enterococci against vancomycin cellular precursor components to decrease the binding of vancomycin. Then Mycobacterium species against the streptomycin. Modification is the modification of the ribosomal proteins of 16S rRNA. Mutations in the RNA polymerase resulting in resistance to the rifampicin kind of antibiotics, and mutations in DNA kinase resulting in the resistance to quinolones. So some examples of bacterial resistance due to drug target modification are also there, like changes in peptidoglycan, changes in other stuff and everything. So these are the four very prominent mechanisms for gaining antibiotic resistance to a drug. So moving on to the next question. Which of the following fecal indicators is used to determine the microbiological safety of water? Anybody? of the following fecal indicators is mostly used to determine the microbiological safety of water? Can you unmute? E. coli. Correct. We'll discuss why it's E. coli. Give me 2 minutes first. There is a lot of noise that's coming. Give me 2 minutes. Yeah. So the correct answer is E. coli. So the correct answer is E. coli, and we'll talk why is it E. coli. So as we know that E. coli is commonly used as a fecal indicator to determine the microbiological safety of water. So the presence of E. coli in water is an indication of fecal contamination and may signal the potential presence of harmful bacteria. Now, viruses and parasites that can cause gastrointestinal illness in humans can also be detected by, E. coli, which is a fecal indicator. So, E. coli is nothing but a bacterium commonly found in the intestines of warm-blooded animals and including humans, and its presence in water indicates the fecal matter has contaminated the water. Thus, monitoring for E. coli, which is a coliform, in water sources is a very common way to determine if the water is safe for human consumption. So the World Health Organization, WHO, recommends the use of E. coli as an indicator organism for assessing the microbiological quality of drinking water. It is a type of bacterium that is very commonly found in the intestines of warm-blooded animals. And although some strains of E. coli are harmless and even beneficial, others can cause serious illnesses such as gastroenteritis, urinary tract infections, and septicemia. So, fecal contamination of water sources can occur through a variety of routes, such as sewage overflow, animal fecal matter runoff, or improperly treated wastewater. And the presence of E. coli in water is an indication that fecal contamination has occurred and other potentially harmful microorganisms might be present. So, testing for E. coli in water is a common way to determine the safety of water for human consumption. And the most common method for detecting E. coli in water is through the use of culture-based methods. This involves taking a water sample and growing it in on a nutrient-rich medium that is selective for E. coli. And if E. coli is present in the water sample, it will grow on the medium and produce characteristic colonies that can be counted and analyzed. And another method for detecting E. coli in water is through the use of molecular methods such as polymerase chain reaction or PCR. And PCR is a very sensitive and specific method for detecting the presence of E. coli DNA in a water sample. So the World Health Organization, or WHO, recommends the use of E. coli as an indicator organism for assessing the microbiological quality of drinking water. And according to them, the drinking water should not contain any detectable levels of E. coli in a 100 ml sample. And in conclusion, E. coli is very commonly used as a fecal indicator. Its presence in water determines that fecal contamination has occurred and the other potential harmful microorganisms may also be present. And testing for E. coli in water is a common method for determining the safety of water for human consumption. And after that, they can probably drink from the same bottle. So these are some of the indicators of the water quality, different indicators. So the thermotolerant coliforms or even E. coli. Some strains of E. coli are very harmless, but some strains can be a little too harsh. So these E. coli are pathogens that help as fecal indicators. So moving on to the next question. Which of the following is the limitation of direct pathogen culturing for detecting pathogenic microorganisms in environmental samples? Anybody? Anybody who's trying to answer? Which of the following is a limitation of direct pathogen culturing for detecting pathogenic microorganisms in environmental samples? So is there any answer? I cannot see. Anybody? Anybody would like to answer? You can try at least. Yeah. So the answer will be number 2. That is, it can underestimate the actual number of microorganisms present in the sample. So, what do we mean by direct pathogen culturing? So, direct pathogen culturing, or the process of growing microorganisms in a lab setting, is a widely used technique for detecting pathogenic microorganisms in environmental samples. However, there are several limitations to this approach. The one of the limitation will be low sensitivity. So many pathogenic microorganisms are fastidious or slow growing, which means that they may not grow well or at all in any standard lab media. And this can result in false negatives and underestimation of the true pathogen load in the sample. Another reason can be selectivity. So standard laboratory media may not be selective enough to distinguish pathogenic microorganisms from non-pathogenic ones, again leading to false positives. Another reason can be viability. Right. Since microorganisms may not survive in the environment for long periods of time or may be present in a non viable state, it can also lead to false negatives. Another is lack of diversity. So, direct culturing may only detect a limited number of microorganisms present in a sample, potentially missing important pathogens that are present in low abundance or are very difficult to culture. And the other last last point will be time consuming. So culturing microorganisms can take several days or even weeks, which can delay the detection and the identification of the pathogens in the environmental samples. So in summary, while direct pathogen culturing is a valuable tool for detecting pathogenic microorganisms in environmental samples, it has still a lot of limitations that must be considered when interpreting results. So that is why pathogen culturing is very it's a very prominent technique that is used in labs all over the world. But still, like sorry, but still, direct pathogen culturing can have a lot of limitations, as I already explained, with respect to underestimation of the actual number of microorganisms present in the sample. So, does anybody have any question with this doubt with this question, or we can move on? Okay, so that means you've understood. So this direct pathogen culturing can very easily underestimate the actual number of microorganisms in the sample, and that's one of the limitations of this technique. Moving on to the next question. Which of the following is an advantage of using molecular techniques to directly detect pathogens in environmental samples? Anybody? Try at least.
Last option.
Fast detection of pathogens. Anybody else who would like a different, uh, like to choose a different option, or do you think any other option might be true? Yeah, so the answer will be fast detection of pathogens. So molecular techniques are also very sensitive and specific. So that option is not mentioned here, but that would have been a very great answer to this question. It's a very huge advantage of using molecular based methods. So molecular based methods are techniques that detect and identify microorganisms based on their genetic material, okay, rather than the ability to grow in laboratory culture. So very common examples of molecular based methods include PCR. So you can see on the screen, these are some of the very prominent techniques, molecular biology, advanced molecular biology techniques that are used uh, to check for microbes in any kind of environmental sample. So, what is polymerase chain reaction? So, polymerase chain reaction is a technique that will amplify a specific DNA sequence from a sample, allowing for the detection of a specific microorganism or genetic marker. And this can be used to detect pathogens that are present in very low abundance or that are very difficult to culture. Next, we have next generation sequencing. So, NGS is a high throughput sequencing method that can rapidly generate millions of DNA sequences from a sample. And by sequencing all the genetic material in a sample, NGS can actually provide a comprehensive assessment of the microorganisms present, including both culturable and non-culturable species. Then we can talk about FISH, which is the very cute acronym, but the actual word is fluorescent in situ hybridization. So, fluorescent in situ hybridization or FISH is a technique that uses fluorescent probes to bind to specific DNA sequences in a sample, allowing for the identification of specific microorganisms or microbial communities under a microscope. Then we have microarray based methods. Uh, like DNA microarray is falls under this category. So in microarray based assays, what happens is that they use small DNA probes to detect and identify specific organisms in a sample, microorganisms in a sample. And these probes can be designed to target specific genetic markers or whole genomics of an organism. So these are some of the techniques. So what are the basic advantages of molecular based techniques? So molecular based methods have several advantages over direct pathogen culturing, including higher sensitivity, specificity, and the ability to detect non-culturable microorganisms. However, they can also be more expensive and technically challenging to perform because it needs certain kind of expertise, and it can also may require specialized equipment and knowledge. So direct pathogen culturing can only detect viable microorganisms, which are those that are capable of growing and dividing in laboratory culture conditions. And if microorganisms are present in a non-viable state, for example, if they have been killed by environmental stressors such as UV radiation, disinfectants, or antibiotics, They will not grow in laboratory media and therefore will not be detected by direct culturing. And it is important to know that not all microorganisms are equally susceptible to these stressors, and some microorganisms can remain viable even under harsh environmental conditions, while others may become non-viable very quickly. So therefore it's possible that direct pathogen culturing may underestimate the total number of microorganisms present in a sample, particularly if non-viable microorganisms are also of interest. And in contrast, molecular based methods such as PCR or metagenomic analysis, metagenomic sequencing can detect both viable and non-viable microorganisms, which can provide a much more comprehensive assessment of the microbial diversity in a sample. So, what is the advantage of molecular based techniques? So, molecular techniques have several advantages over direct pathogen culturing for detecting pathogens in environmental samples. Uh, as I said, there will be high sensitivity. They will be highly sensitive, can detect pathogens at very low concentrations, even if they are present in small numbers in a complex environmental matrix. Also, they will be highly specific. Molecular techniques will be highly specific and can distinguish between closely related microorganisms or strains of the same microorganism, allowing for the accurate identification of pathogens. It can help in rapid detection. So, molecular techniques can provide results within hours to days, depending on the method used, compared to several days or weeks required for culturing. Then molecular based technologies will help in detection of non-culturable microorganisms. So molecular techniques can detect non-culturable microorganisms which may be present in the environment in dormant or non-viable state and are not detected by direct culturing. Then we have high throughput uh, advantage. Molecular techniques can analyze multiple samples simultaneously, making them ideal for large scale screening or surveillance programs. And also culture independent. Molecular techniques do not require culturing of any kind or culturing of microorganisms in a lab, which can be challenging for some fastidious or slow growing pathogens. So, moving on, overall, as summary, molecular techniques offer several advantages over direct pathogen culturing for the detection of pathogens in environmental samples and are becoming increasingly popular for their ability to provide rapid and more accurate information about microbial diversity and pathogen detection. But yes, there are some limitations to molecular based techniques for pathogen detection as well. There is a chance of false positive. So, molecular based techniques can be sensitive enough to detect even trace amounts of microbial DNA or RNA, which can lead to false positives if the detected genetic material does not correspond to a viable or infectious microorganism. Also, there's chances of contamination. So, molecular based techniques can be susceptible to contamination from environmental or laboratory sources, which can lead to false positives or inaccurate results. High cost, definitely, it's not cheap at all. Some molecular based techniques can be more expensive than direct culturing methods, particularly if the specialized equipment or reagents are required. Technical expertise and knowledge, definitely. So molecular based techniques will require any kind of specialized technical expertise and training, particularly for the analysis and interpretation of complex sequencing data, and obviously limited ability to culture microorganisms. So while molecular based techniques can detect non-culturable microorganisms, they do not provide information on the basically on the viability or culturability of detected microorganisms, which may be important for assessing the risk of infection or disease transmission. Also, limited availability of reference databases. The accuracy of molecular based techniques depends on the availability and accuracy of reference databases for pathogen identification, and the completeness of these data sets can vary depending on the microorganisms of interest and the geographical location. So even though these molecular based techniques have a lot of advantage, there are several limitations as well, and their effectiveness for pathogen detection depends on the specific application, the microorganism of interest, and the availability of resources and expertise. And it is important to carefully evaluate the advantages and the limitations of different detection methods when selecting, an appropriate strategy for pathogen detection in environmental samples. So if you can talk about all these techniques individually, conventional PCR or conventional polymerase chain reaction is a molecular based technique that can be used for detection of pathogens in environmental samples. And some of the advantages of conventional PCR might be sensitivity. I've already talked about it. It is very highly sensitive, can detect very low concentrations of DNA, even when they're present in very small numbers. Obviously specific, it's highly specific and can distinguish between closely related microorganisms or strains of the same microorganism, allowing for the accurate identification of pathogens. Speed. So conventional PCR can provide results within hours, making it much faster than culturing methods that can take several days to weeks. Then high throughput. Obviously, it can analyze multiple samples simultaneously, making it ideal for large-scale screening or surveillance programs. Also, quantification. Conventional PCR can be used to quantify the amount of target DNA in a sample, providing information on the concentration of the pathogen present. So these are some of the advantages of conventional PCR, but there are limitations as well. So there is a limitation of there is a chance of false positive. So it can produce false positives due to the presence of non-specific amplification products or contamination during sample preparation. Also, inhibitors. Conventional PCR can be inhibited by substances present in the environmental sample, such as humic acid, which can reduce the sensitivity of the assay. Also, there is a need for a pure DNA template. Conventional PCR requires a pure DNA template for amplification, which can be challenging when working with complex environmental samples that may contain inhibitors or other interfering substances. Also, there is very limited information. Conventional PCR provides limited information about the viability or culturability of detected microorganisms, which may be important for assessing the risk of infection or disease transmission. Then, lack of strain differentiation. Conventional PCR may not be able to differentiate between different strains or subtypes of a pathogen, which may be important for epidemiological investigations. So overall, conventional PCR has several advantages for the detection of pathogens in environmental samples, but it also has some limitations that should be taken into consideration when selecting a detection method. Then we'll go into multiplex PCR. So in multiplex PCR, it is used to detect multiple pathogens in a single reaction, and the advantage is obviously efficiency. Like multiplex PCR can detect multiple pathogens in a single reaction, making it a time efficient and cost effective method for screening large numbers of samples. Then sensitivity. Multiplex PCR is highly sensitive and can detect very low concentrations of target DNA, allowing for the detection of pathogens even when they are present in small numbers in environmental samples. Then obviously specificity. Multiplex PCR is highly specific and can distinguish between closely related microorganisms or strains of the same microorganism, allowing for the accurate identification of multiple pathogens simultaneously. Obviously quantification. So multiplex PCR can be used to quantify the amount of target DNA in a sample, providing information on the concentration of the pathogens present. It is also a very high throughput. Multiplex PCR can analyze multiple samples simultaneously, making it ideal for large-scale screening or surveillance programs. But there are also a number of limitations of the multiplex PCR because it has limited number of targets. So multiplex PCR can be limited by the number of targets that can be detected in a single reaction, which may not be sufficient for certain applications. Also, there is an increased risk of false positives. Multiplex PCR can increase the risk of false positives due to the potential for cross reactivity between different primers or application products. Then multiplex PCR can be inhibited by substances present in environmental samples such as humic acid, which can reduce the sensitivity of the assay. Also, there is a need for a pure DNA template in multiplex PCR for amplification, which can be challenging when working with complex environmental samples that may contain inhibitors or other interfering substances. Also, there is a lack of strain differentiation. Multiplex PCR may not be able to differentiate between different strains or subtypes of pathogen, which may be really important for epidemiological investigations. So overall, multiplex PCR also has a lot of advantages, but so also some limitations that should be taken into consideration when selecting the detection method. Then we can come to real-time PCR. So in real-time PCR, it's a technique for the detection and quantification of pathogens in environmental samples, obviously, and the advantage can be sensitivity. It's very highly sensitive, can detect very low concentrations of target DNA. Specificity, same like conventional PCR, highly specific and can distinguish between closely related microorganisms or strains of the same microorganism, uh, allowing for the accurate identification of pathogens. Speed also, real-time PCR provides rapid results with detection and quantification of target DNA occurring during the amplification process in real time, enabling the detection of pathogens within hours. Then quantification. So real-time PCR can quantify the amount of target DNA in a sample, providing information on the concentration of the pathogens present. And it's very high throughput. Like real-time PCR can analyze multiple samples simultaneously, making it ideal for large-scale screening or surveillance programs. Then what are the limitations? So the cost is a limitation. Real-time PCR is much more expensive than conventional PCR, and the cost per reaction increases with the number of targets in the samples that are being analyzed. Then there's obviously a need for specialized equipment. Like real-time PCR requires specialized equipment including a thermal cycler with fluorescence detection capability, which may not be available in all the laboratories. Then there's a chance for false positives. So real-time PCR can produce false positives due to the presence of non-specific amplification products or contamination during sample preparation. Also, there's a chance there's inhibitors. Real-time PCR can be inhibited by substances present in environmental samples such as humic acid, which can reduce the sensitivity of the assay. Then obviously there is limited information topic that comes into play. So real-time PCR provides very limited information about the viability or the cultural ability of the detected microorganisms, which may be important for assessing the risk of infection or the disease transmitted. So these are some of the all in all some of the advantages and the real limitations of real-time PCR. Then we can go into next generation sequencing. So next generation sequencing is a very high throughput molecular based technique that can be used for the detection and identification of pathogens in environmental samples. The advantage can be that is very high throughput. It can analyze multiple samples simultaneously, making it ideal for large scale screening or surveillance programs. It is also very sensitive, highly sensitive. I can detect very low concentrations of target DNA, allowing for the detection of pathogens even when they are present in small numbers in environmental samples. Also, specificity. It's highly specific and can distinguish between closely related microorganisms or strains of the same microorganism, allowing for the accurate identification of pathogens. Then comes the topic of multiplexing. Uh, NGS can detect and identify multiple pathogens in a single assay, making it a very highly efficient and cost effective method for screening large numbers of samples. Also, whole genome sequencing. NGS can provide whole genome sequencing data for detected pathogens, which can provide valuable information for epidemiological studies and outbreak investigations. But also, as we're discussing for all the techniques, it has also a number of limitations. It's much more expensive than other molecular based techniques such as PCR, and it can limit its use in resource limited settings. Also, data analysis. It generates large amounts of data that requires specialized bioinformatic analysis softwares, which may require additional expertise and resources. Also, limited template quality is another factor. It requires high quality DNA template for analysis, which can be challenging when working with complex environmental samples that may contain inhibitors or other interfering substances. There is a chance of false positives because NGS can produce false positives due to contamination during sample preparation or sequencing library construction. and the turnaround time. So NGS requires actually a longer turnaround time compared to the other molecular based techniques such as PCR, which may not be really ideal for time sensitive applications. So overall, these were some of the applications and limitations of next generation sequencing. Next, we can move on to this FISH, or fluorescence in situ hybridization. See, it's a molecular based technique that can be used for the detection and identification of environmental samples, and the advantages are that it gives rapid results. So fish can produce results in as little as 2 to 4 hours, making it ideal for time sensing applications. Then specificity. So fish is highly specific and can distinguish between closely related microorganisms or strains of the same microorganism, allowing for the accurate identification of pathogens. Next is a visual detection. Fish produces a visual signal which allows for the direct detection of the target microorganisms without the need for any kind of specialized equipment. Then there is an option for multiplexing. So fish can detect and identify multiple pathogens in a single assay, making it a highly efficient and cost effective method for screening large numbers of samples. Then obviously quantification. Fish can quantify the amount of target DNA in a sample, providing information on the concentration of the pathogens present. Regarding the limitations, limited target detection. The fish may not be able to detect all pathogens in a sample, as it relies on the availability of specific probes for the target organisms. Also, there is a huge chance of false negatives, because fish can produce false negatives if the target microorganisms are not actively dividing, or if the probes do not hybridize with the target DNA. And also there is a need for specialized equipment. Fish require specialized equipment such as fluorescence microscope, which may not be available in all laboratories all around the world. Also limited sample size. Fish is limited to small sample sizes, typically even less than 10 ml. And limited throughput. So fish has limited throughput compared to other molecular based technologies such as PCR and NGS, making it less suitable for large scale screening and surveillance programs. So these were some of the advantages and disadvantages of the fish technique. The last technique that we will focus on will be immunological methods. So immunological methods such as enzyme linked immunosorbent assays, or ELISA, and lateral flow assays, or LFA, can be used for the detection and identification of pathogens in environmental samples. So the advantage is that we get rapid results. Immunological methods can produce results in as little as 10 to 30 minutes, making them ideal for time sensitive applications. Also specificity. Immunological methods are highly specific and can distinguish between closely related microorganisms or strains of the same microorganism, allowing for the accurate identification of pathogens. There is a chance for visual detection. So immunological methods produce a visual signal which allows for the direct detection of the target microorganism without the need for any kind of specialized equipment. And it's very easy to use. Like immunological methods are easy to use and require minimal training, making them ideal for use in resource limited settings. And multiplexing. So some immunological methods such as multiplex ELISA can detect and identify multiple pathogens in a single assay, making them highly efficient and cost effective for screening large numbers of samples. If we talk about the limitations, it's very sensitivity is an issue. These methods may not be as sensitive as other molecular based techniques such as PCR or next generation sequencing, and they may not be able to detect low concentrations of the pathogens in environmental samples. Also, there are chances of cross reactivity. Serological methods may produce false positives due to cross reactivity with non target microorganisms or substances in the sample. Then there is limited target detection. Immunological methods may not be able to detect all pathogens in a sample, as they rely on the availability of specific antibodies for the target organisms. Then there is a point of shelf life. Immunological methods have a limited shelf life and may require specialized storage conditions to maintain their effectiveness. And also limited throughput. Immunological methods have limited throughput compared to other molecular based techniques such as PCR and NGS, making them less suitable for large scale screening or surveillance programs. So overall, immunological methods have several advantages for the detection and identification of pathogens in environmental samples, but they also have some limitations that should be taken into consideration when selecting a detection method. Now I'll talk about a technique called FISH, which is something that is very fascinating, at least for me, and I think it might be a new term for many people uh, out there. So FISH basically means a fluorescence in situ hybridization. It is a technique used to visualize and map the genetic material, which is the DNA or RNA, in cells and tissues, and it basically involves the use of fluorescent probes that are designed to bind to specific regions of the target genetic material, allowing for the direct detection of specific DNA or RNA sequences within the cell or the tissue sample. Now, the probes used in FISH are typically short strands of DNA or RNA that are labeled with fluorescent molecules. So, when the probes bind to their complementary sequences in the target genetic material, the fluorescent signal is emitted and can be visualized using a fluorescence microscope. And FISH can be used then to detect specific genes or chromosomal abnormalities, to map the location of genes on a chromosome within chromosomes, and to identify microorganisms in environmental samples. And it is a very valuable tool in molecular biology research and is widely used in medical diagnostics, genetic counseling, and cancer research. So these are some of the techniques that can be used in place of direct pathogen culturing. Some of the molecular biology based techniques to identify and detect pathogens in environmental samples. Anybody has any doubt regarding this question? This a lengthy one. Any doubt? Okay, maybe we can move to the next question. Which of the following techniques is used to assess the viability of bacterial cells in the environmental samples? Anybody?
Last option.
Both EMA and PMA treatments. Okay. Anybody else? Okay. So you're right. And before discussing, we'll talk a little bit about what ethidium monoazide and propydium monoazide are. So I am pretty sure you must have heard about ethidium bromide, propydium iodide. So these are ethidium bromide is very frequently and very commonly used in agarose electrophoresis. What it does is it's basically a dye that goes and intercalates the base pairs of the DNA, stacks itself in the basically intercalates in the base stacks of the DNA, and then you can actually visualize it as a band on agarose electrophoresis. Propydium iodide also does similar things. It is also popularly used in apoptosis experiments or FACS experiments. Basically, propydium iodide will not enter the cell on its own, but if allowed to, if the cells are not permeable or if they are tight, then it will easily enter the cells. So that is one of the concepts that FACS experiments apoptosis assays use. But what are ethidium monoazide and propydium monoazide? So, ethidium monoazide and propydium monoazide are both chemical agents that are used to selectively inhibit the amplification of DNA from a dead or non-viable microorganisms during molecular-based detection methods such as PCR or qPCR. So, the use of EMA or PMA treatment can improve the accuracy and sensitivity of the microbial detection by preventing the amplification of DNA from dead or non-viable microorganisms that may be present in the sample. Okay, so the chemicals work by penetrating the cell membrane of the dead or the non-viable microorganisms and intercalating with the DNA, which makes the DNA inaccessible to amplification during PCR or qPCR. So the EMA or PMA treated samples, as you can see in this picture, are then subjected to DNA extraction and subsequently molecular detection methods which selectively amplify DNA from viable microorganisms. And this approach can help to reduce false positive results and increase the specificity and accuracy of the microbial detection assay. And EMA and PMA treatments are most commonly used in environmental microbiology, food microbiology, clinical microbiology, where accurate detection of viable microorganisms is critical for the identification of infectious agents and the prevention of disease transmission. So, EMA and PMA treatments are used to assess the viability of bacterial cells in an environmental sample by selectively inhibiting the amplification of DNA from dead or non-viable cells during molecular-based detection methods such as PCR and qPCR. And when EMA and PMA is added to a sample containing both viable and non-viable bacterial cells, it penetrates the cell membrane of the non-viable cells and binds to their DNA, making it inaccessible to amplification during PCR or qPCR. And in contrast, the DNA from viable cells remains intact and accessible for amplification. Now, after the PMA or the EMA treatment, the sample is subjected to DNA extraction and subsequent molecular detection methods, which selectively amplify DNA from viable cells. And this approach can help to reduce false positive results and increase the specificity and accuracy of the microbial detection assay. Now, therefore, by comparing the results obtained with and without EMA or PMA treatment, it is possible to determine the proportion of viable cells in the sample, and this information is valuable for assessing the potential impact of environmental factors such as pollutants or biocides on bacterial viability, and for evaluating the efficacy of disinfection or preservation treatments. And these two chemical agents are very popularly used. They have similar mechanisms of action, but there are also some differences between them. So the similarities will be that both are intercalating dyes that can penetrate the cell membrane of dead or non-viable microorganisms and bind to their DNA. Both EMA and PMA can selectively inhibit the application of DNA from dead or non-viable microorganisms while allowing the application of DNA from viable microorganisms. Both EMA and PMA treatments can help to reduce false positive results and increase the specificity and accuracy of microbial detection assays. Then what are the differences? So EMA has a lower affinity for DNA than PMA, which means that it may not completely penetrate the cell membrane of dead or non-viable microorganisms and may be less effective in inhibiting the application of DNA from non-viable cells. PMA is more photostable than EMA, which means that it is less susceptible to degradation from light exposure and can be used with longer exposure times during light treatment to improve the effectiveness of the treatment. EMA is less toxic than PMA, which makes it a safer alternative for some applications. EMA can be used to treat both DNA and RNA samples, while PMA is only effective on DNA. In summary, both EMA and PMA are very useful tools for selectively inhibiting the amplification of DNA from dead or non-viable microorganisms in molecular-based detection methods. And while they share some similarities in their mechanisms of action, there are also some differences in their effectiveness, stability, toxicity, and compatibility with different sample types. And the choice of which treatment to use will depend on the specific needs of the experimental application. Okay. So in recent years, by coupling photoreactive DNA intercalating dyes, biomolecular techniques such as PCR and recombinase aided amplification have been widely applied. And in this approach, they use this dye, ethidium monoazide and propidium monoazide, and they are only used to penetrate non-viable cells with compromised membranes, whereas viable bacteria with intact membrane should pose a barrier for DNA intercalating dyes. And that is how this entire technique is moving forward, and it is quite famous. So this technique, this schematic shows the structures of both ethidium monoazide and propidium monoazide. It's basically the same, only the chain length at one part is different. And this schematic illustration of the working principle of bacterial viability is in the combination of PME-EMA staining and with polymerase chain reaction. The red star stands for PMA or EMA. So, in case of dead cells, the membrane will be compromised, and PMA-EMA will get easy access to the DNA. They'll bind to the DNA, and when PCR happens, this DNA will not be accessible to the primers and everything, so this part of the DNA will not be amplified. But in case of viable cells, the membrane is intact, the dyes cannot get inside or bind to the DNA, and hence the intact cells will show PCR and the amplification will occur. That's the basic protocol of EMA PMA technique. Moving on to the next question. How can the problem of biological instability in environmental samples be addressed during transport? Anybody? Anybody? How can the problem of biological instability in environmental samples be addressed during transport?
All of the above.
Okay. Anybody else? This is a very huge problem regarding biological instability in environmental samples whenever people are shifting samples or transporting from one place to the other. This can be a very, very important thing. Yeah, it's true, all of the above. So the environment that a clinical sample is exposed to is a very important pre-analytical factor in laboratory testing. So there are numerous environmental conditions that specimen may experience before it arrives at the clinical laboratory for analysis. Specimens collected at off-site locations are typically stored at the site and transported to the clinical laboratory via courier, and depending on the geographic location, season, method of storage and the method of transport, the specimen can experience varying climate conditions that can lead to inaccurate test results. The specimens collected within the healthcare institution are not exempt from suboptimal storage and transport environments. For example, specimens that are being transported via pneumatic tube systems can experience extreme agitation and rapid acceleration and decelerations. Suboptimal storage and transport temperatures occur less frequently within health systems due to multiple regulatory requirements for temperature monitoring. However, temperature monitoring may not occur at every step of the pre-analytical phase. So there are multiple reviews online that are talking about the internal and the external environmental conditions that can cause pre-analytical errors in clinical laboratory testing, and what are the strategies that can be used to mitigate environmentally induced pre-analytical errors and regulatory gaps for environmental monitoring in the pre-analytical phase. So there are a lot of factors because specimen transport is a universal step in the pre-analytical phase of the total testing process, and specimens may be collected from outpatient clinics, and collection sites might be hundreds of kilometers away from the clinical laboratory where they will be analyzed. So in the case of reference laboratory, specimens may be transported from different states or countries for analysis. Within the context of a hospital system, specimens may be transported between campuses, buildings, and different areas of the same building. Finally, once in the core laboratory, specimens may be transported by hand or by means of automation for analysis. So, while it is widely appreciated that the environment specimens are exposed to can significantly impact specimen integrity and consequently had lead to inaccurate results, environmental conditions are not routinely monitored, and that is why it's very important to do so. Like climate and microclimate climates can influence environmental conditions very easily that biological specimens experience during transport. There are five major climate zones that include tropical, dry, mild, continental, and polar, with approximately 30 subclimates. And in addition to the major climate zones, microclimates are climate conditions that exist a few meters above or below the Earth's surface. So they occur within each of the five major climate zones, and conditions depend on factors such as temperature, humidity, wind, turbulence, dew, frost, heat balance, and evaporation. And they are further influenced by topographical features within the region, such as lakes, vegetation, as well as large metropolitan cities. And for example, roads, bridges, buildings, and other city infrastructures were found to absorb, the sun's heat within an urban area that resulted in a higher daytime and nighttime temperatures than in outlying areas. So all this is called a heat island effect. And currently, research predicts that this phenomena to increase as the structure, spatial extent, and population density of the urban areas change and grow. And since many central clinical laboratories are in urban areas, microclimates and their impact can, have a major standing in understanding, major consideration in understanding specimen transport. So these are some of the like the use of external specimen transport will continue to expand and grow in complexity as laboratories increase their outreach services. Like specimens commonly originate at an off-site collection facility where patients are being seen for clinical care or phlebotomy. Collection centers affiliated with the hospital, private or reference labs, and specimens may also be collected while patients are being expedited to the hospital via hospital ambulance or helicopter during an emergency, pre-hospital emergency medical situation. And these different modes of transport are subject to unique environmental conditions, including temperature, humidity, storage, and agitation that are not commonly monitored. And each specimen type, be it blood, plasma, serum, urine, tissue, may be impacted differently or not at all by its environment. And in addition, while some analytes require ambient and frozen temperatures, other analytes must be kept refrigerated to ensure specimen stability. And this is known as cold chain transport, and deviation from these temperatures can lead to specimen degradation that can decrease result accuracy. So, unfortunately, maintenance of cold chain transport conditions can be challenging during shipping delays, long distance travel, and underdeveloped city infrastructures. So, these are some of the points that should be kept in mind while transporting elements. Moving on to the next question. Statement A: ingestion of contaminated food or water is the main mode of transmission of Giardia lamblia to humans. This protozoan organism is generally found in water sources, including ponds, streams, and rivers, as well as in polluted food such as raw meat. The statement B is inhalation of polluted aerosols is the main mode of transmission of Legionella pneumophila to humans. This bacterium is usually observed in water sources, including cooling towers, hot tubs, and decorative fountains. Which of the following options do you think is true? Anybody? Which of the options do you think is true?
Both are true.
Okay. Anybody else would like to try? Yeah. So statement A and statement B both are true. So let's talk about the first statement first, Giardia lamblia. So Giardia, the main mode of transmission in case of Giardia lamblia to humans is through ingestion of contaminated water or food. Giardia lamblia is nothing but a protozoan parasite that can cause gastrointestinal illness in humans and animals, and it's found worldwide. And the parasite is shed in the feces of infected individuals or animals and can contaminate water sources, food, and surfaces. So, when contaminated water or food is ingested, the Giardia parasites can infect the small intestine and cause symptoms such as diarrhea, abdominal cramps, bloating, and nausea. And the parasite can also be spread through person to person contact, particularly in settings such as daycare centers, nursing homes, and other institutional settings. Prevention measures include practicing good hygiene, such as washing hands regularly, boiling or filtering water when in doubt of its cleanliness, avoiding consuming food or drinks from uncertain sources, and avoiding close contact with infected individuals or animals. And transmission of Giardia lamblia to humans occur through the fecal oral route, as you can see in the image over here, which means that the parasite is ingested when contaminated fecal matter enters the mouth. And this can happen in several ways, like it can come from contaminated water, like Giardia lamblia can survive in water for weeks to months and is highly resistant to chlorination and other disinfection methods that are commonly used to treat drinking water. And when water sources such as lakes and rivers and wells are contaminated with fecal matter containing the parasite, people who drink the water can become infected. It can come from contaminated food. So the parasite can also be transmitted through the consumption of contaminated foods such as raw or undercooked meat, uh, produce or dairy products that have been exposed to contaminated water or have been handled by infected infected individuals. They can be person to person contact, like Giardia lamblia can be transmitted from person to person through fecal oral route, such as when an infected person doesn't wash their hands properly after using the bathroom and then prepares food or touches surfaces that others will come into contact with. They can be animal to person contact, like Giardia lamblia can also be transmitted from infected animals to humans through contact with their feces or by ingesting contaminated food or water. And once ingested, the Giardia parasites travel to the small intestine where they attach themselves to the intestinal wall and begin to multiply. And this can lead to inflammation and damage to the lining of the small intestine, which can result in symptoms such as diarrhea, abdominal cramps, bloating, and nausea. And the prevention measures include practicing very good hygiene, such as washing hands regularly, boiling or filtering water when in doubt of its cleanliness, and avoiding or consuming food or drinks from any uncertain sources, and avoiding close contact with infected individuals or animals. The first statement is true. Next, we'll talk about the Legionella pneumophila organism. Is basically a type of bacterium that is commonly found in freshwater environments such as lakes, rivers, and streams. However, it can also be present in man-made water systems such as cooling towers, hot water tanks, and plumbing systems, where it can grow and spread if conditions are favorable. So Legionella pneumophila is known to thrive in warm, stagnant water environments, particularly between temperatures of 20 to 45 degrees Celsius. And this is why man-made water systems such as cooling towers and hot water tanks can be particularly conducive to the growth and spread of the bacteria if not properly maintained and clean. And people can become infected with Legionella pneumophila by inhaling aerosolized water droplets containing the bacteria, such as those produced by cooling towers, shower heads, hot tubs, and fountains. And the bacteria do not spread from person to person, and infection occurs only through exposure to contaminated water sources. And Legionella pneumophila can cause a severe form of pneumonia known as Legionnaire's disease, as well as a milder form of the disease called Pontiac fever. So the symptoms of Legionnaire's disease include fever, chills, cough, muscle aches, and shortness of breath, and can be fatal in some cases. Pontiac fever is a self-limiting illness that causes fever, headache, and muscle aches, and typically resolves within few days without treatment. So to prevent Legionella pneumophila infection, it is important to properly maintain and clean man-made water systems, especially those that involve warm water. So this may involve regular cleaning and disinfection as well as appropriate temperature and chemical control measures. Now, the main mode of transmission of Legionella pneumophila to humans is through inhalation of contaminated water droplets or mist. And this can happen when people breathe in small droplets of water that contain the bacteria, such as those produced by cooling tower, hot tub, shower, or fountain. And the bacteria can then multiply and thrive in wild, warm water environments such as found in large plumbing systems and decorative fountains as well. So when the water is aerosolized or broken into small droplets, the bacteria can be released into the air and inhaled into the lungs. And it's important to note that Legionella pneumophila is not spread from person to person, and it is not contracted by drinking contaminated water or touching contaminated surfaces. The bacteria can only cause infection when they are inhaled into the lungs. And prevention measures include a proper maintenance and disinfection of water systems, regular cleaning of hot tubs and spas, and monitoring of water quality in large buildings and other man-made water systems. People who are at a higher risk of developing Legionnaires disease, such as older adults, smokers, and people with very weakened immune systems, should also take extra precautions such as avoiding high-risk water sources and seeking medical attention promptly if they develop any kind of symptom. So yes, both these options statements are true. She was correct. Moving on to the next question. Sorry, what is the primary function of a leachate? I guess that's the pronunciation. Collection system in a landfill. Anybody? Sorry, did you say anything? Okay, it's very self-explanatory. The option 4 will be the correct answer, to collect and remove liquid leachate from the landfill. So the primary function of a leachate collection system from a landfill is to prevent leachate, and it is basically a liquid that forms when water comes into contact with waste and from escaping the landfill and contaminating the surrounding environment. So this collection system typically consists of a series of pipes and liners that are installed at the base of the landfill to collect the leachate, and that is generated by the waste, and the collected leachate is then transported to a treatment facility for proper disposal. And by preventing the release of leachate into the environment, the leachate collection system helps to protect our groundwater and surface water from contamination, and helps to ensure the safety and health of the nearby communities. Okay, so that is what earlier should. So landfills are basically sites for disposal of solid waste. So when water is dumped in landfills, it undergoes decomposition due to the action of microorganisms. It produces a liquid called leachate. Leachate is a highly contaminated liquid that can pollute soil and water resources if left untreated. So to prevent contamination, leachate must be collected and treated before being discharged. And the process of leachate collection generally occurs in a number of steps. First, there is a liner installation. So a liner is installed at the base and the sides of the landfill to prevent leachate from seeping into the soil. And the liner is made up of impermeable materials such as clay or synthetic materials. Then there is a leachate collection system installation. A network of pipes is installed on the top of the liner to collect the leachate, and the pipes are usually made up of high density polyethylene HDPE and are perforated to allow the leachate to enter. Then there is a leachate collection sump. Basically some construction. The collection system pipes are connected to a sump which is largely underground tank that collects the leachate, and then there will be leachate treatment. The collected leachate is then treated to remove contaminants before being discharged in the environment. So a landfill site with an impermeable liner installed at the base and sides, a network of pipes made up of high density polyethylene and perforated to allow leachate to enter, and a leachate collection sump to store the collected leachate, and leachate treatment system to treat the leachate. These are the requirements. Now, what is the advantage? It prevents contamination of soil and water sources, definitely. It reduces the risk of environmental pollution. It helps to comply with regulatory requirements. The disadvantages can be expensive, the collection and treatment. It requires continuous maintenance to ensure proper functioning. If improperly designed installed, it can actually lead to leachate collection system can actually lead to environmental pollution. So these are some of the disadvantages, limitations of the collection system. Moving on to the next question. Carbon dioxide is a product of fermentation of organic waste in a landfill. Fermentation is the initial step in decomposing organic matter in a landfill, where organic complex organic compounds are broken down into similar compounds such as carbon dioxide, organic acids, and alcohol. Methane is produced during the later stages of decomposition through methanogenesis. Hydrogen and sulfide are not typically produced during the fermentation of organic waste in a landfill. Which of the following options do you think is true? Anybody?
Both are true.
Okay. Anybody else in having any different opinion?
Both are true.
Yeah. Yeah, so A and B are both true statements. So we know that fermentation is an initial step in the decomposition of organic matter in a landfill, and when organic materials such as food waste, yard waste, and paper products are buried in a landfill, they begin to decompose due to the presence of bacteria and other microorganisms. And this decomposition process can be broken down into several stages, with fermentation being one of the earliest stages. So, during fermentation, organic materials are broken down by bacteria in the absence of oxygen, and a process called anaerobic digestion. And this produces various organic compounds such as organic acids, alcohols, and gases, including methane and carbon dioxide. Okay, and these compounds are then further broken down by other microorganisms in the landfill, eventually producing simpler compounds such as water, carbon dioxide, and other gases. So, the entire decomposition process in a landfill can take many years depending on various factors, such as the type and amount of organic material present, the temperature and moisture conditions in the landfill, and the presence of oxygen and other nutrients. Now, methane is produced during the later stages of decomposition, especially during the methanogenesis stage. This is basically a biological process that occurs when certain types of microorganisms called methanogens break down organic matter in the absence of oxygen, a process known as anaerobic digestion. And others said that during the initial stages of decomposition, such as fermentation, organic matter is broken down into simpler compounds such as organic acids, alcohols, and carbon dioxide. And as the decomposition process continues, these compounds are further broken down by other microorganisms, eventually producing simpler compounds such as water, carbon dioxide, and hydrogen gas. Now, methanogens then use the hydrogen and the carbon dioxide produced during early stages of decomposition to produce methane gas. And the production of methane gas typically occurs in the deeper layers of the field, where oxygen is limited or absent, and where the conditions are favorable for methanogenic microorganisms to survive. And overall, methane production is a natural part of the decomposition process in landfills and other anaerobic environments where organic matter is present. However, because methane is a potent greenhouse gas, it is important to manage and capture methane emissions from landfills to mitigate their impact on the environment. So you were right. Both the options are true. Regarding hydrogen and sulfide, so hydrogen and sulfide can be produced during the fermentation stage of organic waste decomposition in a landfill, although the production is generally limited compared to other compounds such as organic acids, alcohols, and gases like methane and carbon dioxide. And during fermentation, the organic materials are broken down by the bacteria in the absence of oxygen, producing various organic compounds, including organic acids and alcohols. And these compounds can then be further broken down by other microorganisms in the landfill, producing gases such as methane, carbon dioxide, and hydrogen. So, sulfur reducing bacteria can also be present in the landfills and can produce hydrogen sulfide gas during the decomposition of organic matter. And however, hydrogen land sulfide production is generally more prevalent in later stages of decomposition when the availability of oxygen is limited and the anaerobic conditions become more widespread. So, the production of hydrogen sulfide in landfills is influenced by several factors, including the types of organic waste present, the moisture content, and the temperature of the landfill. In well-managed landfills, the production of hydrogen and sulfide can be minimized through measures such as proper waste management and gas capture systems. So that was the question. So anybody has any questions regarding this? She was correct. The answer will be both are true. Okay. So moving to the next question, which of the following is responsible for reducing methane and carbon dioxide emissions in a sanitary landfill? Anybody?
Anaerobic microorganisms.
Anybody else? Yeah. So you're correct. So when when organic waste is is sent to landfill, it undergoes a biological decomposition process, and the decomposition process is carried out by microorganisms such as bacteria, fungi, and protozoa. So the process includes several stages, including hydrolysis, acidogenesis, acetogenesis, and methanogenesis. And during the hydrolysis stage, large organic molecules are broken down into smaller molecules such as sugars, amino acids, and the fatty acids, and the microorganisms that are involved in this stage secrete enzymes that break down the organic matter. And in the acidogenesis stage, basically the smaller molecules, as I was showing here, the smaller molecules produced in the hydrolysis stage are further broken down into organic acids, alcohols, and carbon dioxide. This stage is carried out by the acidogenic bacteria. In the genesis stage, the organic acids and alcohols produced in the acidogenesis stage are converted to acetic acid, carbon dioxide, and hydrogen gas, and this stage is carried out by acidogenic bacteria. And in the final stage, methanogenesis, the acetic acid and hydrogen gas produced in the acidogenesis stage are converted into methane gas and carbon dioxide, and this stage is carried out by methanogenic bacteria. So the methane gas produced during the decomposing process can be captured and used as a source of energy. And however, if the landfill is not properly managed, methane gas can escape into the atmosphere, contributing to the greenhouse gas emissions and climate change. Overall, the biological decomposition process in a landfill is important because it helps to break down organic waste and reduce the volume of waste in the landfill. However, it is important to remember that managing landfills properly is very important to minimize the environmental impact of the decomposition process. Now, what was this question? Methane and carbon dioxide gas. So, methane and carbon dioxide emission is basically in a sanitary landfill are primarily produced by the decomposition of organic matter that is present in the landfill. And the decomposition process is carried out by microorganisms such as bacteria and fungi that break down the organic matter through a series of biological processes. Now, during the decomposition process, organic matter is first broken down into smaller molecules by a process called hydrolysis. This is followed by acidogenesis, in which the smaller molecules are converted into organic acids, alcohols, and carbon dioxide. Next, acidogenesis occurs, in which the organic acids and alcohols are converted into acetic acid, carbon dioxide, and hydrogen gas. Finally, methanogenesis occurs, in which the acetic acid and the hydrogen gas are converted into methane gas and carbon dioxide. And methane is a very potent greenhouse gas, and its emissions from landfills contribute significantly to global warming. Now, carbon dioxide is also a greenhouse gas, but its contribution to global warming is less significant than methane. However, large amounts of carbon dioxide emissions from landfills can still have a very negative impact on the environment and contribute to climate change. To reduce methane and carbon dioxide emissions from landfills, proper management is necessary. So this includes measures used as covering the landfill with a layer of soil, using landfill gas collection systems to capture methane gas, and implementing waste diversion programs to uh, reduce the amount of organic waste that is sent to the landfill in the first place. So yes, anaerobic microorganisms are primarily responsible for producing methane and carbon dioxide emissions in a sanitary landfill. Those anaerobic microorganisms are those that can survive and carry out their metabolic process without oxygen. Now, in the absence of oxygen, organic matter in the landfill is decomposed by anaerobic microorganisms, leading to the production of methane and carbon dioxide as byproducts of the decomposition process. Now, methanogenic bacteria are specifically responsible for producing methane gas in a landfill, and these bacteria convert acetate, hydrogen, and carbon dioxide into methane gas and carbon dioxide through the process of methanogenesis. And other anaerobic microorganisms such as astrogenic and acetogenic bacteria are also involved in the decomposition process and contribute to the production of organic acids, alcohols, and other compounds that are further metabolized by methanogenic bacteria. So proper management of landfill conditions can help to control the activity of anaerobic microorganisms and reduce methane and carbon dioxide emissions. For example, covering the landfill with a layer of soil can help to limit the amount of oxygen that reaches the waste material, creating conditions that are less favorable for aerobic microorganisms and promoting anaerobic conditions. Additionally, implementing a landfill gas collection system can help to capture and utilize methane gas that is produced during the decomposition process, reducing the amount of greenhouse gas emissions that are released into the atmosphere. So that is what methane and carbon dioxide gas will do in the long run. So anybody has any questions till now? We'll move to the next question then. Give me 2 minutes. I'll come. One second. I'll come.
Option 1.
Yeah. I'll discuss. One second. Yeah, I've already seen it. Yeah, sorry. There was some network laptop issue from my side. Yes. So you're correct. It's option one. Uh, which of the following common characteristic is a common characteristic in old landfills and new landfills? So one common characteristics of old landfills as compared to new landfills is that they typically lack proper engineering and environmental controls. So, in the past, landfills were often constructed without liners or covers to prevent glaciate, that is, liquid that has passed through garbage, from seeping into the surrounding soil and groundwater. So, this can result in contamination of the nearby water resources and the environment. Additionally, older landfills may not have been designed to collect and treat methane gas, which is produced by the decomposition of organic waste in landfills. And this can lead to the release of methane into the atmosphere, contributing to greenhouse gas emissions and climate change. In contrast, newer landfills are typically designed with liners and covers as well as systems to collect and treat leachate and methane gas. So, these measures help to minimize environmental impacts and ensure the safe disposal of waste. And lower levels of methane production in landfills can help to reduce the environmental impacts associated with landfill operations. Methane is a very potent greenhouse gas that contributes to climate change, and reducing its emission from landfills can actually help mitigate this impact. However, it's also important to note that reducing methane production is just one aspect of proper landfill management. Other measures, such as proper siting and design, and effective waste separation and recycling programs, and safe and efficient waste handling and disposal are also very important to minimize the environmental impacts of landfill operations. Moreover, it may not be possible to completely eliminate methane production in landfills, as it is a natural byproduct of organic waste decomposition. And in such cases, landfill operators may need to focus on capturing and treating the methane gas produced in order to minimize its release into the atmosphere. So yeah, you were right. Landfill is a common characteristic of all landfills. Moving on to the so this is how the landfill system can be shown in a schematic format. Moving on to the next question. Which of the following is not a potential application of thermophilic microorganisms? Anybody?
Fermentation of beer and wine.
Anybody else? So that means the other three options are most likely true. So anybody else? Anybody else? Yeah. So yes, you were right. It's fermentation of beer and wine. So thermophilic organisms can be used to produce biofuels, definitely, as you can see in the first option over here, because they are capable of thriving in high temperature environments and are therefore used in various biotechnological processes, including the production of biofuels. And one example is the use of thermophilic bacteria and archaea in the production of biogas. Biogas is a renewable energy source that is produced through the anaerobic digestion of organic materials such as agricultural waste, sewage, and food waste. And thermophilic microorganisms can break down organic materials more efficiently at high temperatures, resulting in higher biogas yields. And in addition to biogas, thermophilic organisms can also be used to produce biofuels such as ethanol and butanol through a process called consolidated bioprocessing. And consolidated bioprocessing involves the simultaneous breakdown of biomass and the conversion of the resulting sugars into biofuels by a single microorganism. And thermophilic bacteria and fungi are most commonly used in consolidated bioprocessing processes because they can tolerate high temperatures and are capable of breaking down complex biomass. Overall, the use of thermophilic organisms in biofuel production is an active area of research and has the potential to contribute to the development of sustainable and environmentally friendly sources of energy. Next, yes, thermophilic organisms can be used to help in the bioremediation of contaminated soils. Bioremediation is basically the use of microorganisms to break down and remove or transform contaminants from soil, water, or other environments. And thermophilic microorganisms are particularly useful in bioremediation of contaminated soils as they can tolerate higher temperatures and are capable of breaking down complex organic compounds. And in addition, thermophilic microorganisms are known to produce more enzymes than mesophilic organisms, which thrive at moderate temperatures, which can enhance their ability to break down contaminants. So, thermophilic microorganisms have been used successfully in the bioremediation of soil contaminated with various organic compounds such as petroleum hydrocarbons and polycyclic and aromatic hydrocarbons. The thermophilic bacteria and fungi used in this process break down the contaminants through the process of aerobic or anaerobic digestion. However, it is important to know that the effectiveness of bioremediation using thermophilic microorganisms can depend on various factors, such as the type and concentration of the contaminants, the temperature and the moisture levels of the soil, and the availability of nutrients for the microorganisms. Therefore, site specific conditions must be evaluated before implementing bioremediation to thermophilic microorganisms using thermophilic microorganisms. Also, thermophilic microorganisms play an essential role in composting of organic waste. Composting is a process that involves the controlled biological degradation of organic materials into a nutrient rich soil amendment. And thermophilic microorganisms are particularly important in the high temperature or hot phase of composting, which occurs after the initial mesophilic or warm phase. And during the hot phase, the temperature of the compost pile can reach up to almost 70 degrees Celsius, which is the optimal temperature range for the growth and activity of thermophilic microorganisms. And thermophilic microorganisms break down complex organic compounds into simpler compounds such as carbon dioxide, water, and humus, which are very readily available to plants and nutrients. And the high temperatures can also help to kill weed seeds, plant pathogens, and other harmful microorganisms that may be present in the organic waste. And thermophilic microorganisms used in composting include bacteria, fungi, and actinomycetes. And these microorganisms produce a variety of enzymes that break down different types of organic compounds in the waste, including carbohydrates, proteins, and lipids. And the breakdown of these compounds generates heat, which further promotes the growth and activity of thermophilic microorganisms. So, in summary, thermophilic microorganisms are essential for the high temperature phase of composting, which is a critical step in the conversion of organic waste into nutrient rich soil amendment. Thermophilic microorganisms can also play a role in the fermentation of beer and wine. However, it's important to know that most beer and wine fermentations are actually carried out by mesophilic microorganisms, which thrive at moderate temperatures. Now, in beer brewing, Saccharomyces cerevisiae is the most commonly used yeast species for fermentation, and it is a mesophilic organism. However, some like especially specialty beer styles such as Belgian saison can be brewed using thermophilic yeast strains such as Saccharomyces dies. Saccharomyces, which can tolerate higher temperatures than Saccharomyces cerevisiae and produce unique flavor and aroma profiles. So, in winemaking, also a variety of microorganisms are involved in the fermentation process, including yeast and bacteria. And the primary yeast species used in wine fermentation is Saccharomyces cerevisiae. Also, it is a mesophilic microorganism. However, some wine producers use thermophilic bacteria such as Oenococcus oeni for malolactic fermentation, which can occur after the primary fermentation and can improve the wine's flavor and stability. In general, while thermophilic microorganisms may not be the primary species used in beer and wine fermentation, they can still play a huge role in the production of certain beer and wine styles or in the specific stages of fermentation process. So even though it does not play a very it's not the primary microorganism, it still can be used for the fermentation of beer and wine. So we'll stop here today. Anybody has any question regarding the question, any doubt regarding the question that we discussed today, or we can stop the session for today. If there is any question. Okay, so I have been uploading all these lectures to YouTube, and I have been uploading the presentations as well. So you can always take a look at it when needed. So if you don't have any question today, maybe we can conclude the session, and we'll meet next Friday with a new set of questions, same link, same time. Thank you so much. We can conclude the meeting. Thank you, Neha, for being so interactive. We can conclude the meeting today.